Good evening, ladies and gentlemen, and welcome to this evening's talk. My name is Paul Nurse. I'm president of the Royal Society, and it's a pleasure to see you here this evening. I'm given instructions to tell you. They're very boring, but I'm told to say them. You've got to turn off your mobile phones. I'm to tell you we're being webcast and recorded. And I'm to tell you that if we have to escape, the best ways are at the back, but you could also try that one over there. I don't think we will have to escape, but just in case you need to. Now, it is a real pleasure for me to introduce tonight's lecture. Dr. Nick Lydon I've known for many years, and I've known of his work for many years before that. He is going to be awarded the Royal Society GlaxoSmithKline Prize, and it involves a lecture that's given once every 2 years, and it's given for original contributions to medical and veterinary sciences. It says published within 10 years from the date of the award, but I think we liberally interpret the 10 years. The prize does consist of a very nice gold medal. And it requires an evening lecture at the Royal Society, and that's what we're celebrating tonight. I should say a little about the prize itself. It was initially established following a donation from the Wellcome Foundation and was first awarded in 1980, the centenary year of the foundation. But since 2002, given all the mergers and so on that has happened, it's been supported by GlaxoSmithKline Limited. And the award this year is to an outstanding scientist. And I think I do not exaggerate, and being British I tend to understate, a scientist who is responsible for a highly significant paradigm shift in how we should think about the development of pharmaceuticals. And the particular drug that this work has generated, Gleevec, Nick played an extremely important role in that development. And it's a drug which conceptually is very exciting, based on knowledge of how a particular disease works, and design of a drug that would target the altered protein that was important for generating that disease. And the diseases, of course, are different cancers, initially CML and then gastrointestinal stromal tumours. And the use of the drug has saved or prolonged the lives of many thousands of patients throughout the world. And it has revolutionized our way of thinking about drugs, as I've said, and it's taken certain diseases, the ones I've just mentioned, into a treatable condition by targeting the oncogene that is the molecular basis of that cancer. These diseases were virtually untreatable in the 1990s, and they've now become extremely important for treatment, and also very, very significant for thinking about future cancer drug discovery. Nick has won a number of international prizes, and I've been entangled with several of them. And in fact, when we last met, he was getting yet another prize. I was very pleased that he got the Lasker Clinical Medical Research Award. I'm on the jury of that, and so knew all about what happened there. And that's often known as the American Nobel Prize in Medicine. So, enough from me. Nick, can I invite you to come and give your lecture?
Thank you. Thank you, Paul, for that very kind introduction. So what I'd like to talk to you tonight about is the kinome. And this represents a large group of homologous enzymes that are known as protein kinases. And the function of these enzymes is central to what we know as signal transduction now. So the talk will introduce you to a little bit of what we mean about signal transduction and oncogenes. And I'm going to take you back to when I started this work at the University of Dundee in the early 80s and what we knew about these particular targets. And then proceed into giving you an overview of what CML is, which is the primary target of a drug known as Imatinib, or its trade name Gleevec. And then in the second part of my talk, I'll tell you a little bit about what we're doing currently at Blueprint Medicines. And hopefully this will lead to the next generation of targeted therapies. So this diagram illustrated signal transduction as we knew it in the early 1980s. And this was fortunate for me because I ended up doing my PhD across the corridor from the lab of Philip Cohen, who was working on this particular signal transduction pathway. And that is the action of glucagon on cells, signaling from the hormones signaling from the exterior of the cell, leading to a signal transduction cascade. And interestingly, these involved protein kinases. And the reversible phosphorylation of these particular enzymes in the pathway led to a change in function which regulated the output, which was liberation of glucose into the circulation in the fasted state or synthesis of glycogen in the fed state. And at that time, when I started at Dundee, there were very few of these protein kinases known. This is the earliest one recognized, the protein kinase A, which I worked on in my PhD. And as you can see, during the early 80s, there was an explosion in discovery of enzymes from this family, the protein kinase family. And this was due to improvements in molecular biology techniques, especially homology cloning using degenerative PCR methods. So here we are today. The full complement of kinases in what we call the kinome are 518, of which 90 are able to phosphorylate. That means the transfer of phosphate, the terminal phosphate from ATP to a substrate containing tyrosine. And what this phosphorylation reaction does, which is common to all these enzymes, is it takes the terminal phosphate, the gamma phosphate from ATP, it adds this to a substrate, and these substrates then have an altered function. So as you can see, this massive explosion in the number of kinases, highly related in their active site, in the early 80s created a problem for us at that time, because how were we able to monitor selectivity of so many enzymes? It also created an opportunity in that we knew these were involved in signal transduction. Therefore, if they became deregulated, they may be applicable as pharmaceutical targets in many different situations. So the other piece of information that was breaking at that time came from the oncogene field, the retroviral oncogene field. And shown here is a schematic of what this involves from a paper from Tony Hunter around about 84, which really got me interested in targeting this class of enzyme with drugs. And the take-home message here is that these very small RNA tumor viruses, which only had 3 genes, when they infected their recipients, sometimes, very rarely, they were able to pick up an altered form of a cellular gene. And these tended to be modified in that the regulatory domain on the N-terminus of the protein was usually removed, and the GAG protein, a structural part of the GAG protein, was added. And the result of this was these oncogenic, viral oncogenic protein kinases had enhanced activity and were able to cause tumors. Now, normally these tumors would have, these rare events would have disappeared, but these viruses were rescued from these tumors, and shown here are a number of them. And when they were sequenced, it was found that they all, or a large proportion of them, had this protein kinase activity within them. And shown here is the simplest form of the protein kinase family, the cyclic AMP-dependent protein kinase. So they all had homology to this domain. So at that time, there was no real connection of these oncogenes or viral oncogenes to human cancer. And that came with the finding that this particular viral oncogene, the V. abelson oncogene, was highly related to the first gene that was linked as a genetic abnormality in cancer. And this came from a number of contributions, starting with Nolan Hungerford, who were able to identify this chromosome, which is now known as the Philadelphia chromosome. And this was present in almost all cases of chronic myelogenous leukemia, which we'll refer to as CML. So what happens in CML is there is what's known as a chromosomal translocation between the long arm of chromosome 9 and 22, which results in this abnormal chromosome seen by Nolan Hungerford. And it wasn't until 74 when the late Janet Rowley was able to determine by high-resolution chromosome banding that this what this translocation resulted in was the result of the Philadelphia chromosome. And then subsequent work from a number of groups that worked out the molecular consequences of this translocation. So what happened, these two chromosomes, 22 and 9, at the fusion point, you create a hybrid messenger RNA, a hybrid gene, and the product of this is the BCR Abelson protein shown down here. And very similar situation in the viral V. abelson gene. The N-terminus of the protein is removed and an unrelated sequence is added, which leads to deregulation of the kinase activity and high enzyme activity. So what does this do? This Philadelphia chromosome translocation occurs in a hematopoietic stem cell located in the bone marrow. Normally, this is the site of stem cells which create all the cells which circulate in the periphery involved in immune defense. For example, the myeloid side, granulocytes, monocytes, and then the lymphoid side, T cells and B cells. So when this translocation occurs in a single cell, it gives it proliferative advantage, and gradually you get an expansion of the myeloid stem cells carrying the Philadelphia chromosome. This is shown here, and this is known as the chronic phase of the disease. And there's a gradual increase in these red cells, which are circulating blast cells, which are found in the periphery. But at a certain point, this chronic leukemia transforms into an acute leukemia, and there's another genetic event which leads to what's known as blast crisis. And this is invariably fatal. There's a massive expansion in circulating blasts. And interestingly, recent information has suggested that the defect, or one of the transforming events which results in blast crisis, is another kinase known as GSK3, which incidentally was one of those I showed you on that signal transduction slide from Phil Cohen's early work. So this summarizes the clinical course of CML, starting in the chronic phase, which can last from 5 to 6 years. You then go into an accelerated phase during transformation, and then a very short fatal blast crisis. And when you look down here, this shows the blood cell counts from these different phases. So in normal patients, the white blood cells are less than 10,000 per cubic millimeter, and there's a massive expansion of the myeloid compartment in chronic phase. And as you develop blast crisis, there's an increase in the number of circulating blast cells in the periphery in the bone marrow. So what were we trying to do? When I started at Ciba-Geigy in 1985, along with Alex Matter, the concept was to find a key which fitted into this kinase. And we can visualize each of these members of the kinase family, these 500 kinases, as being like a lock. And to get a selective drug that only inhibited that particular kinase and not all the others, we needed to sort of understand what the key looked like to fit in this lock. Obviously, there's a master key that goes into every kinase. That's its substrate ATP. And that was the basis for starting. So it's a laborious task at that point of screening for what we call lead compounds that we can then chemically modify to dial in selectivity for its target. So this particular slide summarizes many years of work, and it started in 1990. We'd been screening for kinase inhibitors for quite a while, but never really came up with a compelling chemical series that had drug-like properties. So in 1990, this scaffold was identified in a screen, not for Abelson, for a related enzyme we were interested in called protein kinase C. And during the modification of this scaffold, the decoration of this scaffold by Jörg Zimmermann in the chemistry department, we were able to come up with this final molecule, which is in fact Gleevec. And the breakthrough for us really was the addition of this simple flag methyl group to the molecule, which changed its specificity from being an inhibitor of serine threonine kinases to being a relatively selective inhibitor of what we were able to identify at that time, 3 major targets: the Abelson kinase, and then 2 other protein kinases, the PDGF receptor and the c-KIT receptor. This had all the properties we wanted in a molecule, and I've summarized a few here which were an early challenge with, which were good solubility and very good oral bioavailability in rodents. And this shows you some of the data from back in the early 90s. So as I said, there were 500 plus kinases in the kinome. We only had expressed and purified around about 30 of these kinases. And so we knew it had selectivity, but we really didn't know the full picture of its specificity. And in the cell-based assays we had established at that time, it was able to inhibit both the V-Abelson kinase and the BCR-Abelson kinase in cells, and similar concentration. It was also able to block PDGF receptor signaling and c-KIT signaling as monitored by phosphorylation of the receptor. And it showed specificity with respect to a number of other targets in cell-based assays. Jumping forward to what we know now, this is an assay I'm going to talk about a little bit later. It's now commercialized by DiscoverX and enables us to look at the specificity of kinase inhibitors across the whole kinome. And you can see on this dendrogram, each of these little nodes represents an individual kinase. So the green, the blue circles represent the targets that it hits: PDGF, KIT, Abelson, and its related ARG. And then the red ones were kinases we discovered after the event were also modulated. The size of the circle represents the potency of the drug against that target. And that can be seen here in a more easy view of potency along the bottom against the different targets. And this is the Abelson kinase. So the question we had at that time was, what was the molecular basis for the selectivity? Because we couldn't interpret the structure-activity relationships from chemistry across this compound class with its actual specificity. And it wasn't until John Kurian published the crystal structure of the Abelson kinase bound to Gleevec that this became apparent. So shown here is the Abelson kinase in its 2 activation states, shown here inactive and then active form. And these two forms are obviously regulated very tightly in the normal Abelson kinase. In the BCR Abelson, they are stuck in the on position, so it's always phosphorylating its target substrate. As you can see, there's a lot of movement in the protein around this, what's called in the activation loop, which is what regulates access to the active site. Shown on this side is, in yellow, Gleevec binding to the inactive form of this kinase, and in white, the natural substrate for the kinase ATP. And as you can see in this representation, the Abelson kinase, the Gleevec binding to the Abelson kinase accesses a pocket at the back of the enzyme which is only present in the inactive form of the kinase. And it's a very different location than what is seen for ATP. Although ATP, the adenine and the head group of the inhibitor occupy the same space, the tail of these molecules occupies a very different location in the active site. And as you can see in this inactive form, ATP is incompatible with binding because there's a steric clash with this activation loop. And that really explained to us why this compound is so selective for these kinases, which from an examination of their sequence would not suggest that they would be the same, would be all inhibited by this one drug. And this is data from Dan Trieber at DiscoverX showing what happens with Gleevec binding to these two forms of the enzyme. Shown in white is the active form, and shown in black is the inactive form. And this is binding data rather than biochemical. And as you can see, the affinity of binding to the inactive form is far more potent than that to the active form. Whereas other inhibitors, such as dasatinib, which is a second generation kinase inhibitor targeting the Src and Abelson kinase, has equipotent binding to both forms of this enzyme. So what does this do in the cell? In a collaboration with a number of labs, Carlo Gambacorti in Milan and the Taupaz group in MD Anderson, we screened through as many CML and ALL cell lines as we could find. And the CML cell lines are shown here. They all have the P210 version of the BCR Abelson gene. In Philadelphia positive cases of ALL, that's another leukemia, there's also another form of Abelson, a slightly shorter form, and all of these were exquisitely sensitive to killing by low concentrations of Imatinib. We took other leukemias, which were Philadelphia chromosome negative. There was absolutely no activity of the drug against these leukemias in cell culture. In addition, we had cell systems involving the 32D cell line, which is the IL-3 dependent cell line. And if we transform these with P210 BCR Abelson, we are able to convert them into factor-independent cells, which grow in the absence of ligand, shown here. Now, if we add in Gleevec in the presence or absence of IL-3, we can completely block growth of these model cell systems. And this type of system became a workhorse for many of the assays we currently use today. In addition to that, we were able to monitor phosphorylation of the BCR Abelson in this cell line and shown that at concentrations that blocked proliferation, it also blocked the primary target. Autophosphorylation of the Abelson gene had no effect on IL-3 dependent signaling through the JAK2 kinase. Now, for those of you in the pharmaceutical industry, that is really where a lot of the hard work starts. And what I'm not going to talk about is many of the activities required to get that from the discovery phase through into the early clinical development stage. And what I'm going to mention is what I think was really important for this project, which was the translational medicine biomarker aspect of the development. And that was done in collaboration with Brian Drucker, who I initially met while working with Tom Roberts at Harvard Medical School on developing very early in this program tools that we could use to express protein kinases. And Brian had developed the first monoclonal antibody against phosphotyrosine, which we've used, or the industry used subsequently for almost all studies in monitoring tyrosine phosphorylation in cell cultures. And when he had moved to OSHU, he called me up and he knew we were working on Abelson inhibitors, and he was looking to do translational work and clinical development. So really important to us, in which were experiments that convinced Ciba-Geigy at that time, which subsequently became Novartis, to actually invest in this program, were a set of experiments he did which really, I think, validated in everybody's mind that this small indication, which marketing department was not interested in, would be a good testing point for concept validation of protein kinase inhibitors in cancer. And what he had was patients, both normal and CML, where we took bone marrow or blood and then incubated these in methylcellulose in the presence of various growth factors that lead to outgrowth of precursors of the granulocyte, the myeloid lineage, or in this case, the erythroid lineage. And shown here is what happens in the presence of Gleevec if you take these cells from normal subjects. Colony formation for the blast-forming or the colony-forming GM lineages, you can see is not affected by the presence of the drug. However, if you add Gleevec, you get a dramatic reduction in colonies, both erythroid and GM, in the presence of the drug. But importantly, when you pick these colonies and analyze them for the presence of the BCR-Abelson oncogene, you can see that only a minority of them express the BCR-Abelson mRNA. Whereas the input cells prior to treatment were 99% positive for BCR-Abelson translocation. And what this told us, that Imatinib is able to select for growth of the normal hematopoietic progenitor cells out of this population of overwhelmingly BCR-positive cells. In addition to that, from when we took patient samples and blotted them for phosphotyrosine, these are the white blood cells from CML patients, 2 patients, you can see you could, at 1 micromolar Imatinib, you could completely block this aberrant signaling monitored through the phosphorylation of cellular proteins on tyrosine. And this again was using this monoclonal antibody that Brian Drucker had developed. And interestingly, in the translational medicine part of this, we were able to monitor one of these phosphorylation events. This is of a downstream substrate of Abelson called CRKL in patients as we increased the dose of Gleevec, and were able to determine a effective dose for target engagement in the early phase 1 studies. So moving on to the phase 1 study, this represents the dramatic effects we saw very early in the phase 1 study. And as the dose of Gleevec was increased in patients, this represents the 500 milligram per day group, but at doses above 300, you saw dramatic normalization of the white cell count. So this is referred to as a hematological response. And the normal range for most people in the room will be in the range 5,000 to 10,000 per cubic mil. You can see in this particular patient, that is dramatically elevated to over 100,000 white blood cells per mil. And these normalized very rapidly over the course of 30 days. And this was sustained over greater than a 1 month period. And what was very interesting from the translational medicine perspective is the dose we'd predicted, the plasma trough levels, at the dose we saw these responses was around about 1 micromolar. So that validated the translational models we'd been using to determine a dose in the clinic. Now, in addition to these hematological responses, which were observed at doses above 300 in almost all patients except one, there were also what we call cytogenetic responses. That means disappearance of the Philadelphia chromosome in the metaphase of samples taken from the bone marrow of these patients. And in addition, there was one patient that had a complete molecular response, meaning you couldn't determine BCR Abelson expression by the most sensitive method at that time, which was qPCR. Now I show you just this slide to give an idea of the timelines and how rapidly this occurred after getting into clinic. So although it was first synthesized in '92, it took us quite a while to get into clinic due to a few preclinical hiccups. But once it started in the phase 1 study in June '98, it was rapidly approved within 32 months after dosing. Now, what was the impact on CML as a disease? Now, this shows the incidence shown in blue in the US, the yearly incidence of the disease of roughly 4,000 cases. And as you can see, after the introduction of Imatinib, the mortality from this disease dramatically declined. But what is not shown here is the increase in prevalence over time has increased dramatically with the decrease in mortality. Fortunately for patients in this group, there are now second and third generation inhibitors that address some of the reasons for relapse in this particular group, and I'll touch on that briefly. So, following the publication of that that phase 1 clinical trial, this appeared on the cover of Time, and obviously hyping the fact that there was new ammunition against these cancers. And these were the magic bullets. But unfortunately, as in many situations, like in use of antibiotics and bacteria, selective pressure on these highly proliferative diseases lead to escape mutagenesis. And this summarizes work from the Sawyer's group, who were able very early to detect mutations in patients that relapsed under Imatinib therapy. And shown here are the particular mutations. There's roughly 12 clinically relevant positions in the protein that becomes mutated, and this results in resistance to Imatinib therapy. Now, I mentioned, fortunately for the patients, there are second-generation inhibitors that are approved. One, nilotinib, a close structural analog of Imatinib with higher potency against the primary target and also higher potency against some of these mutants. And a less selective compound, dasatinib, which also has a very high potency against the Abelson kinase. And this summarizes some of these second generation and now third generation compounds, which are able to even modulate the escape mutants, which for so long haven't been inhibited. That's the T315 to I mutation, which was the most resistant to therapy. So in the introduction, Paul mentioned GIST. So this was not a disease we intended to target, and it was an activity of Gleevec we were very worried about because we thought it would come along with significant toxicity to normal hematopoiesis. Around about the time when we started the phase 1 clinical studies of Gleevec, a publication from a Japanese group showed that this particular target, the c-KIT gene, was mutated in I think 5 out of 6 patients with GIST that they looked at. And this led to rapid clinical trials in gastrointestinal tumours and the high activity of the molecule compared to previous treatments. This was a solid tumour as opposed to a chronic leukemia, and it was refractory to all known chemotherapy and radiation. So this was really an indication to us that these signal transduction molecules could also be effective in solid tumours. I'm going to mention this disease a little bit later. This is systemic mastocytosis. Gleevec was also active in rare forms of this disease which did not contain a particular mutant known as D816V. This is resistant to Gleevec, but the rare cases which had activated KIT, which didn't involve this particular mutation, Gleevec was highly effective. And I'll come back to that in the second part of my talk. So this paradigm shift that Paul mentioned earlier really involved a new way of looking at cancer drug discovery, which was to understand the disease and specifically the pathogenesis, what the molecular defects, what the targets we should go after, the driver mutations in these cancers were, and then design a targeted drug as selectively as possible that could be address this specific molecular abnormality in the cancer. So moving forward to around about the 2005, while I was at a meeting, I met 2 scientists, Patrick Zarenka and Dan Treba, who had developed an assay methodology for looking at the specificity of kinases. And it was a very clever trick by expressing kinases as a fusion with a phage, then to barcode that and develop a binding assay where this particular kinase could bind to an immobilized inhibitor. And then you can compete this with a drug, wash away, and using quantitative PCR, you could measure the concentration of the kinase that was retained on the bead. Now, this enabled us to do massive parallel assays by mixing these kinases. So you could multiplex this, and it really gave a good way of doing kinase specificity in a very simple format. It didn't involve ATP, so every kinase was measured apples to apples rather than the presence of its cofactor. And as I showed you earlier, we can generate these dendrograms of specificity. At their current format, they have 464 kinases in this particular assay. Now, this assay now is commercialized by a company called DiscoverX, and I will come to how we use that later. But what it enabled us to do is give each kinase inhibitor a real selectivity measurement. And shown here are a number of kinase inhibitors that were available around 2010, and some of these were FDA approved, and we gave them a selectivity coefficient. That is the number of kinases hit over the number of kinases measured. And as you can see, the most selective compounds are at this end, and the least selective, such as the natural product staurosporin, which inhibits almost every, or binds to almost every kinase in the kinome. And you can set the concentration of the drug you want to measure this selectivity index. And at Ambid, we used this to develop a very selective FLT3 inhibitor called AC220, which is currently in clinical trials, phase 3 clinical trials for AML, a different type of leukemia, which has a deregulated FLT3 receptor. And shown here is this exquisitely selective compound which hits only 2 or 3 related receptors in this family. Now, this was really helpful in the medicinal chemistry. You can see here compounds against kinases, and we were able to monitor its true selectivity against all the targets in the kinome simultaneously on a real-time basis as we made compounds. And this enabled us to generate these dendrograms, and here I just showed a number of approved compounds in contrast to AC220. It has a selectivity similar to Imatinib, whereas some of the broader spectrum inhibitors that are used clinically tend to hit quite a number of kinases in the kinome. So around about 3 years ago, along with Alexis Borosy, my longtime collaborator Brian Drucker and Charles Sawyer, we started Blueprint Medicines, aimed at taking advantage of the technology advances such as the DiscoverX kinase assay and advances in genomics that have been occurring at rapid speed. And we believe the time is right to merge those two, both in identifying novel targets in both cancer and other diseases, and developing very selective kinase inhibitors against these targets. So the idea is here. We wanted to develop compounds that look like this on the dendrogram, very selective. And you might ask, well, some of those previous compounds I showed you are in clinical use and are effective. Why not have broad spectrum kinase inhibitors? So the reason for that I've listed here. We want to be able to dose these kinase inhibitors at very high levels to get full inhibition of the target, to prevent both escape mutations occurring and to minimize the toxicities involved in off-target inhibition of kinases that you're not targeting. And why we think this is incredibly important is we know that escape mutagenesis is going to be the major clinical problem with kinases. So these drugs will undoubtedly, especially in the solid tumors, have to be combined with other molecules. So to prevent overlapping toxicities, we want these to be highly selective with as minimum number of off-target toxicities as possible. And this also allows you to do very early clinical proof of concept with a selective drug against a genomically defined patient population. So what we've developed at Blueprint is two platforms. One is the synthesis and annotation of a novel kinase library, and I'll speak to that next. And the other is a genomics platform where we look for novel targets at the genetic level. And what we're interested in is the overlap between these two particular platforms. So on the library side, many of you know that there are many kinase libraries out there that have been synthesized, are commercially available. Many pharmaceutical companies have synthesized many kinase inhibitors. So what we did was a slightly different approach. We selected a number of scaffolds, such as the starting scaffold I showed you earlier for Gleevec. And then we differentially decorated them, and each of these scaffolds slightly differently vectors substituents into different part of the active site of this kinase family. They're all synthesized with drug-like properties, and we were not interested in inhibiting a specific kinase. We were completely agnostic. And then we screened all these kinases through the entire kinome and looked for their selectivity. And we did this iteratively until currently we have about 10,000 compounds. And this shows a sort of schematic of what we do. So kinases down this side, compounds across here, and using this heat map, we can see different patterns. The ones which hit many kinases, for example, looking this way, we're not really interested in. Those that have very selective patterns, for example, hitting a number of kinases, we do another iteration of chemical synthesis. The current result of this is we have high quality medicinal chemistry starting points against probably 80% of the kinome. 60% of the kinome is covered with 3 or more scaffold, 40% with 6 or more scaffolds. And out of these, we've selected what we call tool compounds. These are very selective compounds, and we're able to inhibit with these compounds roughly 250 different kinases at present. So the other part of the program is target discovery. And we have a number of platforms running to be able to do that. And I'm going to talk about 2 of those. Fusion protein discovery, that is kinases that are fused with other genes, similar to the situation with BCR-ABLsm. And I'll also talk about one aspect of the phenotypic screening we're doing with these tool compounds. So the phenotypic screening is a collaboration with the Sanger Institute, Alton McDermott and Matt Garnett. And this looks at cancer cell line selectivity against a selection of our tool compounds. In this first iteration, we took 70 tool compounds which targeted novel kinases of unknown biology. That means we don't know what they do. And the particular assay format is to run them through their 1,000 plus cancer cell lines and do IC50s for these drugs. And then the ones that have interesting profiles, we can look at the genetic characterization of the cells where they were sensitive, since they have complete genetic annotation for their cell panel. So this shows the sort of data you get out of one of these screens. And it's a heat map. Potency of the drug is colored at the top. Here are the cell lines. Here are the 70 compounds. And you can see they broke down into a number of different patterns. Kinases inhibit every proliferation of every cell line. Some which inhibit a few, and some which inhibit 1 or 2. And so this sort of gives you a feel for the breakdown of those. 2 of these were broadly killing. 41 were able to kill 2 to 20 cell lines, and some of them were able to only hit 1 cell line. And these are the most interesting ones to us, the middle column. Of course, there were some inactives which probably don't modulate proliferation but have some other function. Then for those cell lines which have genomic features in the current databases, COSMIC and the Cancer Cell Encyclopedia, we can look at those, the cell line annotation versus the compound annotation with respect to their inhibition of the different members of the kinome. And I'll just give you a feel for a few of these. For example, this is a compound which hits only 1 cell line. Here's one that hits 2. Here's one that hits a number of cell lines. And in this particular case, the annotation of mutations matched that of the compound profile. So this was an inhibitor of CSF1 receptor and the PGF receptor, and you can see in both those cell lines they have translocations or mutations activating those particular kinases. And then some have broader profile. But all of them, starting off straight from our chemical library, have potencies in the middle to low nanomolar range in cell-based assays. Moving on to one of the other discovery platforms, this is fusion discovery. And this is an exercise where we're looking for proteins which are deregulated in a similar way to BCR-ABLsm. And there are other examples of that which you probably know about, the ALK fusion, which is the target for crizotinib, another selective kinase inhibitor, which is used in non-small cell lung cancer. And obviously, the viral oncogenes, which were all fusion proteins and activated. And shown here is an example of this. This is the RET oncogene in lung cancer, showing the substitution of this with an N-terminal region that deregulates this kinase. So what did we do? There's a lot of information out there available on genomic profiling of many different cancer types, and this includes RNA-seq data. And so we scientists at Blueprint developed a new search algorithm for rapidly detecting fusion proteins. And this takes the raw sequence data out of the databases, such as the TCGA, aligns them, and then looks for chimeric and split reads amongst the data. Now, this has obviously all been done before by other people, but we were able to isolate a large number of new fusions in solid tumors using this methodology. And I'll give you an example of a few of these. So this is a paper that published last week in Nature Communications, which gives the landscape of fusion proteins in solid tumors by looking at data across all the available RNA-seq data within the TCGA database. And they're coded according to whether they're known fusions, that is, they've appeared in the literature before, whether they're a known kinase but with a new fusion partner, whether they're a known kinase but in a novel indication, or whether they're completely new. And I'll briefly run through these. So these are the known fusions, and it recapitulates what was known out in the literature before. Interesting, we have a group shown here, which is a known kinase that's appeared in a fusion before, but with a new fusion partner. And these are shown here, and some of them have very significant incidence. Shown in purple is a known kinase, but in a novel indication. And these are obviously very interesting to us, that they expand the opportunity for developing an inhibitor against such kinases, because the kinase, although not very high incident in a single tumor, is well represented when you look at pan-tumor instance of the translocation. And then lastly are novel fusions that haven't been detected before. And shown in blue are two which have immediate clinical relevance, because MET mutations are known as transforming. This is a validated oncogene, and yet we see MET fusions in quite a number of cancers. In addition to this, there are MET inhibitors on the market, and so this has immediate clinical impact for diagnosis and also therapy of some tumors. The same goes for PI3C alpha. This is the first publication of fusions involved in this gene, and as you can see, it occurs across a number of cancers. Shown in yellow are those which are more intriguing. FGR is an oncogene that was implicated as a viral oncogene, and so this is rather intriguing because it's the first example of the FGR as an oncogene in humans. So to wrap up, I'm going to go back to a disease, systemic mastocytosis, which we initially thought would be treatable with Gleevec because it was due to activation of KIT. But unfortunately, the majority of the cases have a activation mutation that makes them resistant. And I'll talk about this particular program here, which we hope to have in clinical trials early next year. And just to introduce you to this disease, this is a myeloproliferative disorder which starts with mast cell disease, and here's a photograph of a patient with this particular myeloproliferative disorder. It can vary widely. So here's a diagram of the KIT receptor. As I said earlier, some mutations occur in this domain here. These are sensitive to Gleevec. So we know from a concept validation point of view, if you target this particular receptor in a sensitive subpopulation, then you can treat this disease. But the majority, 95%, have a D816 mutation, which renders them resistant to that particular drug. So this, in a similar way as CML evolves out of a blood stem cell, and involves the mast cell compartment of the hematopoietic system. Now, there are 3 or 4 different forms of this disease. There's the indolent, which these patients have a normal lifespan, and they are seen mostly by allergists. There's the aggressive form of the disease, and then 2 very aggressive forms with associated non-mast cell disease and mast cell leukemia, which is rapidly fatal. So this illustrates some data on the inhibitor we have in development at the moment, which is a very potent inhibitor of the D816 mutant form of the KIT receptor. And as you can see here, this looks at inhibition of tumor growth in the mouse in a xenograft model. It is highly effective and causes no weight loss in the animals, whereas a broad-spectrum kinase inhibitor, although it can modulate the anti-tumor activity in the xenograft, leads to rapid weight loss, as this is above the toxic dose used in humans. And at the same time, we can modulate the target, showing target engagement in the animals during these studies. And this is always a nicer way to look at it. This is a luciferase expressing tumor, and you can see here in a diagrammatic form the appearance of tumor in vehicle, rapidly fatal in this animal model by day 8. Here's dasatinib, a marketed drug, which has some activity on the D816V. Tumors reappear at day 8, whereas with the 30 mg/kg dose group in this particular inhibitor, tumors are delayed and start appearing only very late. So I'm going to wind up now and leave you the message that I think the paradigm that was established by Gleevec has been highly effective in bringing to the forefront targeted therapy. And what we are hoping is the next generation of drugs will have the properties of high selectivity, potency, and ability to combine with other promising drugs that are working their way through the development pipeline, which don't target kinases. And the combination of these, we hope, will be the way to get over one of the biggest problems we see in the horizon, which is resistance to single-agent therapy. So I'd just like to acknowledge some of my collaborators in this work. Firstly, the work on Gleevec, especially Jörg Zimmermann in chemistry, Elizabeth Buchtunger in biology, who were pivotal in all the work I described on Gleevec. My longtime collaborator, Brian Drucker, for all the translational work, and Charles Sawyers, and George Dimitri for the GIST work. And of course, Alex Matter, who gave me the opportunity to do all this work very early in my career at Ciba-Geigy, now Novartis. And then the work at Blueprint, led by Christoph Langauer, our CSO, and the library build work in chemistry, the collaboration with the Sanger, and the KIT program, the exon 17 inhibitor work shown here. And then lastly, Dan Trieber and Patrick Zarenka, who initially worked with me at Ambit, and Dan now works at DiscoverX, and Patrick is now at Pfizer. With that, I'd like to thank you for your attention.
Happy to have a
yeah. Sorry about the microphone.
Good. Thank you very much, Nick. So we're now open for any questions or comments. And I think we have a microphone. And so at the back.
Yes, thank you for a wonderful talk.
If you could stand, maybe we could
sure.
then... Yeah, thank you.
Thank you for a wonderful talk. My question is in 2 parts. Part 1 is, in the clinical course of the CML disease, what factors would cause it to go from the chronic phase to the blast phase? And the second part of my question is, at what stage do you introduce the Gleevec therapy?
So the first question, the chronic phase is clearly due to a driver event, which is the creation of the BCR Abelson and the Philadelphia chromosome that results from the translocation. That leads to a gradual expansion of a myeloid or tumor stem cell population, which is probably in the myeloid pathway. And what's thought is that the active Abelson oncogene leads to a proliferative advantage for those cells, so they gradually occupy and overgrow the normal bone marrow cells. Within that cell population, there's then either one or two additional mutations which cause a block in terminal differentiation. And that is the stage where you get the rapid blast crisis, appearance of immature blast in the circulation and bone marrow. And the work from Irvin Weissman suggests that maybe deregulation of the beta-catenin pathway through a misspliced form of the GSK beta enzyme, another kinase. So when the activity of that goes down, which is found in stem cell populations, it causes the blast crisis. That's hypothetical, I think, at the moment, but it seems a valid hypothesis. And so the second question was, sorry, I missed
At what
that.
stage do you introduce Gleevec?
You have to introduce these drugs as early possible in the course of the diseases as you can. The experience with Gleevec shows us that you get very excellent clinical activity in early phases of disease. As you treat patients that get into the blast crisis, they relapse relatively rapidly after responding to the initial therapy. So once you've got into blast crisis, the instability of the tumor, additional mutations that occur, it's hard to overcome, which really brings to the forefront the importance of integrating the diagnostics early enough in disease so you can catch these diseases before they're a frank leukemia or an advanced solid tumor.
Thank you. Simon.
That was a brilliant lecture, Nick. A couple of questions. You mentioned screening and then identifying compounds that were selected for a particular kinase. How useful have you found crystal structures in trying to rationalize that selectivity?
Yes, we've in all the chemistry programs, we do iterative crystal structure, and luckily for us, this is a relatively easy thing to do nowadays, since many of them are commercially available. So you can turn around these crystal structures on a useful timeline where you can actually influence the next iteration of chemical synthesis. So we do that extensively.
Okay. And then you talked, it was a brilliant paradigm shift with respect to science and targeting populations, but there's been a paradigm shift with respect to pricing. So, monotherapy is expensive enough, and if we go to combinations, how do you think healthcare systems are going to cope with triple combinations?
Probably they won't. So I think that's a big issue. What you presume, I think, take CML. It's a sort of an outlier because it turned a chronic disease into something that you can probably live with lifelong, and that's driven the incidence of disease dramatically, and turned what was a market projection when I was at Novartis of peak sales of around about $150 million a year to, I think, what was reported in 2011 of $4 billion a year. And that was driven by the increased prevalence because of the drug kept patients alive. I think that's not going to be what's going to happen in many of the advanced cancers. So what's the combination of diagnostics and targeted drugs will reduce the patient size in many of these indications rather than expand it. But definitely some of the newer drugs are extremely highly priced.
So it's lovely talk, thank you. I wanted to go back to the crystal structure, because when you described the initial design of, or discovery of Gleevec, you described it as something that was designed to be similar to ATP, but when you showed the crystal structure, it looked like it was an allosteric fit. So did you design it to the pocket, or did you find out that it was allosteric?
That was all
later on?
retrospective. So when when we were doing this program, crystal structures had just become available. So Susan Taylor and Janusz Spadsky published the cyclic A kinase, and we tried very hard to model this particular compound into the active site. And we actually published a structure where it was completely wrongly fitted because we were using the active conformation. We believed these inhibitors bound to the active conformation. And of course, it couldn't be modeled into the inactive form. And so it was only retrospectively when John Kurian solved the real binding mode of Gleevec that it all became crystal clear why this was such a remarkably sensitive, selective drug.
Good. Maybe the last question at the back.
Hello, thank you very much for the talk. I'm intrigued because I think the Gleevec story is an exquisite story where impeccable science leads to a pharmacological agent which then made billions of dollars for a pharmaceutical company. How much of that money was then given back to the scientists who were fundamental in that discovery? And if you were to do it again, how could the scientists be rewarded more justly or encourage more scientists to do fundamental research?
Hard question to ask, or sort of answer. I think, when you look back to when Gleevec was discovered, I don't think anybody predicted it would have sales of that size based on, its multiple activities against multiple tumors. And as I told you, that the marketing forecasts when we actually put that into clinic were minuscule. And Ciba-Geigy would not have recouped its development costs if that had been accurate. So it sort of puts into perspective, the high risk of developing a drug. And at that point, it was looked at, which was, one of the reasons we had such difficulty getting into clinic was the marketing department were not interested in a small niche disease. And so we had to dress it up as being a bigger market size because it could access the PDGF receptor and the KIT receptor. The reality is many of these drugs fail in clinic. So this is a poster child, but there were probably 12 or 15 other cancer drugs during that time at Novartis which failed and cost an enormous amount of money in development. So when you look at the cost and the reward, you've got to take into consideration that a lot of things fail and they cost also. With respect to reward of scientists, when things are successful, none of us actually make any money in addition to our salaries. So I'm not quite sure what the reward you're meaning would be. I mean, we get publications and credit for the contribution, but if you work in industry, you don't get rewarded additionally. You might get promoted if you're lucky.
Seems the right time to finish the questions.
Thank you.
I'm going to ask Keith Peters. So Keith Peters come to the podium to assist me with awarding the Scroll Medal and check. Keith is the senior consultant for R&D, GSK, and prior to that was, of course, Regius Professor of Physics in the University of Cambridge and is one of our most distinguished clinical researchers in the UK. Keith.
Thank you. So, first of all, thank you for a really splendid lecture. Just to be slightly personal, as you've heard, I was Professor of Physics in the University of Cambridge. Physics, not physics. The physicists were much cleverer. But I qualified as a doctor in 1961. And as a medical student, I remember very clearly the description of the Philadelphia chromosome, which was the basis of the research that you described, which led to this drug. I also reflect that it was 1960, 1980s with the development of oncogenes that you started exploring possible therapeutic approaches to this form of leukemia, so distinctive a leukemia. And then I observed that it was in 2001 that you did your first phase 1 trial. And several years later, you had a very unusual triumph in that you turned a disease which was hitherto untreatable into one which was dramatically nearly cured. I just make these points to emphasize the long timelines between the investment in underpinning science and the benefit to mankind. Now, as you've heard, I'm a senior consultant for GSK, one of the world's largest drug companies, and I've done that for the past 9 years. And it gives me particular pleasure to be here this evening because we very rarely acknowledge the work of scientists in industry. I think you're the first person in the 1918, 34 years that this prize has been awarded. You're the first industrial scientist to get it. And in that time, it's just worth pointing out to fellows of the Royal Society and the President that the pharmaceutical industry has made a big impact on very important diseases. In my lifetime, for example, HIV has been turned from a fatal disease into a chronic, nearly cured disease, and that work was done almost exclusively by the pharmaceutical industry. And just to give another plug, as we speak, West Africa is being ravaged by the Ebola virus, and the only hope, I think, for stopping that is a vaccine. And the only people who are likely to make a vaccine are the pharmaceutical companies that are expert at making vaccines. And that includes GSK, who have a vaccine in phase 1 trials at the moment. So it remains for me, if somebody spoke about reward to industrial scientists, you have a special reward this evening. First of all, you have the distinction of giving this lecture, which you've done brilliantly. And secondly, you have a much more tangible reward in the form of a check, which I'm told is hiding under here. And it's rather an anonymous-looking check, but nevertheless, it's quite substantial. Congratulations, Nick.
Thank you very much.
On behalf of GSK.
And we have some more humble things to give you. A scroll from the Royal Society and a medal. And I just want to show everybody. It's a very nice medal here. Perhaps I shouldn't have fingered it. There we are. So many, many, many congratulations.
Thank you very much. Okay, thank you.
Okay.
Thank you very much, ladies and gentlemen. Thank you, and it was a wonderful lecture for a wonderful scientist. Thank you very much. Thank you, Nick.